I'm very glad to present my work here, and congratulations also to the this conference, online conference of Plant Genome Online 2022, and this is give a new style of plant genomics meeting, and also freely accessible to all the students and the colleagues in this community. So today I want to share with you our work on hybrid potato, and discuss with you on how genomics can enable the green revolution of the most important tuber crop. So I'm working in Agriculture Genomics Institute at Shenzhen, which is one of the 34 institutes of Chinese Academy of Agricultural Sciences. Potato is the most important tuber crop. So is the number 3 stem food after rice and wheat, and is stem food for more than 120 countries and serving more than 1.1 billion people. So it's potato was domesticated in the Andes by Indians, and the domestication of potato reduce the length of stolon and the increased size of tuber. And the wild and primitive cultivated potato have different progeny level. And the modern variety, the majority is tetraploid. There are 2 structural obstacles in the potato breeding industry, and the one is related to its genome is auto-tetraploid. So that means the genome heterozygosity is extremely high, and the genetic analysis are very complicated. Breeding is not accumulative. So the very routine methods like a backcross are not applicable in potato. And second is colonal propagation, and that means a huge carbon footprint in storage and transportation, and also susceptible to disease and pests. So why tetraploid is difficult? Because for a diploid, 2 eggs, and you have 2 gametes and 3 zygotes. For tetraploid, if you consider double reduction, you have 10 gametes and 35 zygotes. That's for a single locus. So this makes the genetic analysis very difficult and breeding very slow. And let's compare the car business with potato breeding. 100 years ago, cars look like this, and now it's very fancy and can be driven without a driver. So Rosetable Bank is a cultivar released in 1902 and remain a major cultivar. So it's still occupying 40% of the potato area in United States and 70% of the processed potato in the country. And so you may think that it has no shortcoming, but it is susceptible to all the major disease. All the major disease, and so still we cannot identify a potato cultivar in this 120 years that we can have a comparable processing quality of this cultivar. Colonal propagation is troublesome. It means it has a low propagation index. You plant 1 tuber, you got 10 to 15 tubers, and also high cost, about 1,000 to 2,000. dollars per hectare for, and this carries tuber-borne disease and pests. So can we make a potato a more breeder-friendly and farmer-affordable crop? And so we, a few years ago, and together, I think, including some teams in Holland and United States. So we start hybrid potato breeding. We think it will be a new green revolution. That consists of two basic changes, and is to transform tetraploid to diploid. That means we develop inbreeding line, make F1, and because that will transform this non-cumulative breeding into faster iterative breeding, and we also transform clonal propagation into true seeds, easy to ship and store, and also to avoid tuber-borne disease and pests. Some of you may never seen the seeds of potato and the fruits. The fruits are like the green fruits, like a wild potato, a tomato, and 1,000 potato seeds is like 1 milliliter. Is like 1 of 6 of the size of sesame seeds. And we know there's enormous potato production potential in Africa, in Southeast Asia, in South Asia during the winter time, because at that time the climate is cool, and in some area they have water access. And so we can create a new landscape of food production with hybrid potato. And it will not doable with the common potato, tetraploid potato, because if they cannot produce seed potato tubers in those areas. So if we want to grow 6 million hectare potato in Africa, and so for seed potato, that means 3 tons per hectare. You need 18 million tons of potato to ship from the cooler region to Africa, and of course, well, it will get rotten during the shipment. Well, by seeds, it's 180 tons of seeds, so it can be carried by a single plane. So how feasible is this endeavor? What are the challenges? Of course, we can learn from the success of hybrid maize. Was evolution between 1910 and to 1930, and which increased the yield for 8 times. It took this 20 years to finish the research and the commercialization, but we know that there's no correlation between ploidy and the yield. Some people may doubt that as the reduce the ploidy level in potato, you get a smaller tuber, but we think that should not be, it's not the fault of the ploidy level, because we know that in the same level of tomato ploidy level, you can increase the fruits for 100 times bigger with selecting only thousands of seed genes. So hybrid potato is of course more challenging than hybrid maize or rice. Hybrid maize was made in the US, and hybrid rice was made by Renlongping and colleagues in China. So because potato is originated from tetraploid to diploid, that's you have to reduce the ploidy level, and it's transformed from asexual to sexual reproduction from tuber to seeds. And we know that in the genome that in the hybrid rice and in the original rice or maize population, large effect of deleterious mutation are not exist, because due to sexual reproduction, and But in potato, there's a lot of large effect. Deleterious mutation exists hidden in the heterozygous status in the genome, and also we know there's a loss of co-linearity between haplotypes. So we break up this job into 5 steps to decode, understand the heterozygous genome genotype, to understand diploid diversity and self, to create self-compatible material, and select paired deleterious allele cross to select the best of one. To decode, so in the year of 2011, we have published the potato genome paper together with 14 countries, 28 institutes, and about 200 scientists from this formed this potato genome sequence consortium. And we sequence a monohaploid with 12 chromosomes and 39,000 genes. And when we sequence that commented by new scientists with the title code "One Genome Unraveled 3 to Go," that remind us that potato is a tetraploid. And but we don't directly go to tetraploid. We sequence the diploid with RH with very high heterozygosity, and so we designed a de novo assembler and phasing for heterozygous diploid genome pipeline, and we know that deleterious mutations dispersed on the 2 haplotypes. So that means meiotic recombination cannot throw out all the deleterious mutations. And the RH have 24 chromosomes with 76,000 genes. And we carried on sequence cooperation 88, which is tetraploid of 48 chromosomes and about 150,000 genes. So again, to sequence the tetraploid is very complicated. And we again developed a protocol to de novo assembler and phasing this auto tetraploid genome. And it's on the how to say, in revision in molecular plants, and you can see how diverged between these 4 haplotypes within a single genome. So with this we can detect which 2 haplotypes are from the father and which 2 are from mother, and we know that the cross of 2 parents to form the original two parents from using 2 gametes, unreduced gametes to produce tetraploid was the origin of tetraploid potato. And why it was selected? Because it was blocked, how to say, blocked deleterious alleles in this from parents. And we also look at the co-linearity loss in potato genome, and we compare with maize. Maize is already a very divergent genome compared with wheat and the rice, and yet is compared with you can align how to say so you can see the difference between between 2 phases, different phases, and potato genome are much more diversified than diverged than maize. And to zoom in chromosome 1, and you can see the rearrangement, structural variation between 2 potato genome and compared with 2 maize genome. So second step is we're going to genotype the diploids, and we have collected, thanks for the contribution of our International Potato Center, we have accumulated diploid material germplasm from them, and so we've about 400 lines. So they look how different are they? And normally we only pick up the, what are familiar with is the up left one, and all others are I think the diversity is hardly used in potato breeding. With sequencing all of them, we have about 64 million SMP identified. And using this data, we confirm Solanum canalinum is the ancestor of cultivated potato. Cultivated potato have different subspecies. And we developed a pan potato genome from this collection with 44 diploid potato genome from the potato tuber bearing clade section potato, including also 2 non-tuber bearing species from neighboring sister clade, Etibrosum. And with the novel sequence, assemble this 46 genomes with very high standards using hi-fi assembly. And we use a multi-omics approach to identify the tuber initiate gene identity, gene called IT1, identity of tuber 1. Is because of the evolution of this gene, and the potato gain the ability to bearing tuber, And we also, this pan genome can create inversion maps, which are we identified about 200 larger size potato inversion in the potato genomes, including a 5.8 megabase inversion, which related to the accumulation of beta carotene in the tuber flesh. So this will provide critical insights for backcrossing breeding. Genome diversity provides raw material. We provide raw material for breeding, and the third step is to self-cross to overcome self-incompatibility, and we applied 3 strategies. One is lock out the SNS using CRISPR-Cas9, and strategy 2 is to integration of S8. S8 is a natural mutation of at SNIs in one of the land races. So strategy number 3 is to apply of SLI. SLI is a S-locase inhibitor gene from wild species, Solanum shakanza. So it is like a master key, can unlock a lot of self-incompatibility in potato. So this gene was also widely used in our colleagues in Holland. We have cloned them. So after overcome the difficulty of self-incompatibility, and we're going to develop inbred lines. So inbred line, so the more difficult one, than the self-incompatibility if it's inbreeding depression. So inbreeding depression is inherent to outcrossing species, but it remains largely unknown at genome level. So inbreeding depression exists in every different phenotype of potato, including yield, tuber yield. However, the tuber mutation burden is line specific. About 11% of the between on average 2 lines, only 11% deleterious mutation are as the same. So they are line specific. So we can use different line to mask the mutation on the other line. Again, we develop a parental independent genotyping by sequencing methods. So we use genome-wide segregation distortion to detect genome-wide segregation distortion, because segregation distortion is always related to large effect deleterious mutation. So we identified major fitness defects. So inbreeding depression can occur at any phenotype and at any stage, including survival, vigor, and fertility. And we also found that, as I said, major deleterious mutations are located with segregation distortion. So with this information, we start to select the starting material. So normally you select the best one to start to develop inbred lines. But I think maybe it's not correct, because we start with the we sequence all the 400 lines. We select the lines with low number of heterozygous SNPs and also low number of genome heterozygosity. So include, for example, these two can be developed as inbred easily, more easily than these two. So one of the lines is called PG6359. It's a diploid, self-compatible, it carries less deleterious mutation, and have yellow flash color, the beta carotene. So we self these lines to map the deleterious mutation. We including fertility and flowering, and also yellow phenotype. We also select the yellow flash beta carotene, high beta carotene contents traits, and also the shape of the tubers. Sometimes then we have to throw out the deleterious mutation and accumulate, stacking the beneficial alleles. But sometimes it's not so easy to throw out the deleterious mutation. Even we know, for example, in one of the area we know that after self generation of selfing, so this area still carries a lot of heterozygosity. So because there are two deleterious mutation are tightly linked in repulsion phase, so they need a crossing over between them to get homozygous. So of course, when this occurred in the telomere region, it's easy to identify a crossing over to break them. But if it's in the centromere region, it will be difficult. Anyway, we finish. We using the genome design, genome selection. We identified. We gradually improve the homozygosity of inbred lines up to 99%, and that's some of the beam maps of F6 plants. And these are I think the performance of F6 inbreeding line generation. Each plants are from a single seed that looks rather uniform. That means the genome homozygosity is high. We develop more inbreeding lines. So number 5 is step 5 is to cross. And that's one of the cross looks like. And this is the female parents, and this is the male parent. This is the F1, and each of them are from a single seed. That looks rather uniform, and of course F1 it has a strong vigor than the parents. And these are few other lines. And this H4 hybrid number 4 looks very promising. Have the dry metal contents about 26%, and carries a lot of beta carotene, 72 milligram per kilogram. It carries more beta carotene than carrots. And the yield potential is like between 35 to 43 tons per hectare. Of course, we did only in a smaller area, but it can further increase because they also bear a lot of tubers, a lot of fruits. If we use male sterline, it will reduce it. So this is we also study the molecular basis of heterosis of hybrids, and we know that was mainly due to dominant complementation. It's no different with rice. So all the systems are developed as a genome design of hybrid potato, including 4 steps: selection of starting material, genetic analysis of F1 S1 population, and the development of embryos with continuous selfing and selection, and also selected 2 parents. This was received some attention from our colleagues, and Cell published a leading edge article on comments on this one, and also our colleague in Germany published a paper on the reinvention of potato. And Sophie Hamon from Salisbury Library commented they essentially reinvented potato from scratch. And it is in Chinese, Yuan Longping is the father of hybrid rice. He thinks they will bring the green revolution of potato. Future direction will be, we were going to develop a new algorithm for genome selection, including using the prediction of better prediction of deleterious mutation from genome, and the non-host resistance to lead blight. We want to develop to really get rid of this pathogen disease. It will, of course, will be not a short-term goal. And breeding for winter season for tropical area, I think we'll change the food supply landscape in the global food security system. And also we want to develop novel food and feed. And of course, we will need to develop a new practice and mechanization with our colleagues, not by ourselves. Because sowing system is much different with sowing the tubers. With this, I will end my talk, and I thank the contribution from my amazing team, and also funding agency from Ministry of Agriculture and Rural Affairs, and Chinese Academy of Agriculture Science, and the Guangdong Provincial and Shenzhen Municipal Government. Thank you. I would like to take some questions.